This program is brought to you by Stanford University. Please visit us at stanford.edu.
So what we're going to do today is I'm going to take you through the analysis that this PhD student of mine, Bill Dean, and I did back almost 35 years ago now that still lives on today as sort of the definitive model of what's called glomerular ultrafiltration in mammalian species. So not only will you be able to see how some of the simple mass balances we've done in this class can be put to good use, you'll also learn something about transport across membranes, which is a topic that pervades chemical engineering and applications, particularly in the areas of separations. And I went over the physiology with you last time, so let me just review that by sketching out our little model glomerulus. So if you're trying to answer the question, what are the determinants of glomerular filtration rate? And that's the question you get asked. How do you apply engineering analysis to come up with a solution to that problem? So we learned last time that we can sort of qualitatively look at the architecture of the system. So this is the first thing you would do when you make a model, is you sort of get a little picture of what this of what the model might look like. So what I'm doing is drawing for you kind of a schematic of the capillary bed known as the glomerulus. And so you can keep things straight here. It's a sort of separating capillaries from one another. So we know that we have capillaries that are running roughly, and we can treat them as in parallel to one another. And if you can get some semblance of that in your notes, the idea would be is that blood is entering a glomerulus through the afferent arteriole. So it's coming in here like this. Here comes the blood. It then goes into all the little capillaries, comes back out. The efferent, you can think of E as exit, efferent arteriole. And then what is surrounding this little pocket of capillaries? You remember from the physiology? So there's the blind end of a little tube that collects what? The filtrate. So I'll draw it like this. And it goes down like this. And since I'm going to need it, I'm going to take it out over here. So the blood gets filtered across these capillary walls, and it's the plasma minus the proteins. The proteins don't go across. So it's things like glucose and the salts cross these capillaries. And they go into this space, which is this white area I've drawn here. And the filtrate begins its travel down what's called the proximal tubule. So this structure that surrounds the glomerulus is called the proximal tubule, and this space into which the filtrate goes is called the Bowman's capsule, named after the person who first identified it histologically. And so now we have sort of a model of what this looks like. We know that there's millions of these glomeruli, but if we could understand how one works, then we might be able to understand how the whole kidney works by just assembling them all together, since they work massively in parallel with one another. So the key thing to remember in your mind is that filtrate, which is formed and is devoid of proteins and red cells, goes into the Bowman's capsule and begins traversing down this tubule structure. Now, downstream of the tubule structure is another capillary system. So these are called glomerular capillaries. And downstream is another capillary bed called the peritubular capillaries. And these peritubular capillaries serve what purpose? What are they doing? They're reabsorbing this fluid back into the systemic circulation, leaving behind what? Waste, which is constituted primarily of nitrogenous species like urea, uric acid, and creatinine, which then on the way make it down to the bladder and then get expelled. So we're looking really at the front end of two capillary systems that are in series, trying to understand what controls this process of filtration. What are the determinants? Because if we understood what controlled it, then we might be able to exert control over this through interventional therapies, medical therapies, to change the rate at which the filtration occurs in patients who need it. So let me show you the experiments that had been done. This fellow we worked with, Barry Brenner, who was at that point in time a young professor at UCSF, had found a species of rats called Munich Wistar rats in Germany when he was there. And he had them shipped to the United States because he found that when you anesthetize the rat and lay it out on a little tiny operating table, and you cut it open, on the surface of the kidney, you'd see these dark spots. And those dark spots were an occasional glomerulus that was very near the surface. And the reason it was dark is because it had so much blood in it. Most of the time when you look at the surface of a mammalian kidney, you don't see that. They're buried beneath what's called the capsule. And so he said, well, gee, if I can get this mutant rat whose glomeruli lie very near the surface of the renal capsule, of the kidney, then I would, since I can see it, maybe I can access these compartments with micropipettes and measure pressures and withdraw fluids and measure concentrations of solutes and be able to study a single glomerulus. And if I can study a single glomerulus, then I can infer what all the other glomeruli are doing as well. So the experiments that he did was, first of all, to take a micropipette. And the way you make a micropipette is you take a piece of hollow glass tube, you hold a flame over it, and you just start to pull as it melts. And as it does, it necks down and finally breaks. And where it breaks, the tip diameter can be as little as 1 or 2 microns. So this gives you essentially a micro syringe that you can then stick in through the surface of the kidney and into these structures. Now you can't hold it in your hand. So these are put into jigs that are controlled almost like a video game, which ratchets down your shaking hand, gears it down so that you can actually control the motion of the pipette and watch it under a microscope, under a light microscope, and drive it into the kidney and into these structures. So he had one set of pipettes, and I'll just draw it like this, where he could measure the pressure in the Bowman space. I'll call it PBS, Bowman space. He can measure the pressure in the Bowman space. The Bowman space, if you think about it, this thing is filled with fluid, so it has a hydrostatic pressure in it. You stick the pipette in there, and the pipette itself is liquid filled. Then if you match the pressure so that no fluid goes out of the pipette or into the pipette, then you're pushing a back pressure on this that you can measure with a set of bellows, and you can actually measure the pressure that's inside here. You can also stick that same pipette right into a capillary like this and measure the pressure in a glomerular capillary, PGC. So he put that instrumentation together and made such measurements. Now, what we're after is trying to determine what controls the glomerular filtration rate in this glomerulus, and we call that rate that we're trying to measure SNGFR, and that means the single nephron glomerular filtration rate, SNGFR, because in fact, we're going to try to measure the rate at which filtration is occurring in a single glomerulus. And in fact, we coined this term, SNGFR, and Barry Brenner had a Porsche. And in the very first year, in 1971, is when California came out with these personalized license plates. And we gave him a license plate for his Porsche, which said SNGFR. It's very cool. I had one too. It was SNGFR exclamation point, because we couldn't have the same license plate. And then Bill Dean put the exclamation point in front on his little VW. But we crashed that into a tree on one of the trips, so that one was taken off the road. Now, what else can he measure? The other thing he measured, which was really very interesting to try to get at SNGFR, how much that is he would put this, you saw it on one of the slides last time, this material called inulin. Inulin is a sugar. And inulin has the property that it's freely filtered across the capillary walls, just like glucose is. But it has a property that it doesn't cross the tubule wall. So any inulin that gets filtered travels all the way and comes out into the bladder. So it can get across the capillaries, but it can't get across these white walls of the proximal tubule. So you can infuse this through an IV into the animal. And you can take a sample of blood, systemic blood, and you can measure the concentration of inulin in the plasma. So these are things you can measure. So this is the plasma concentration of inulin. So you can measure that. The other thing that he can do is, the interesting thing about the proximal tubule is that water can cross it. Inulin can't. So immediately what happens is this filtrate comes into the proximal tubule, and begins to move down the tubule, water is being taken out. But what's happening to the inulin and as it moves along here? What's happening to its concentration? It's going up or down? It's going up because I'm taking water out and the inulin stays behind, so it's getting concentrated. So then what he did is he took, with a micropipette, he puts a drop of oil in this tubule. And then he sucks fluid out of this pipette at a rate that keeps this droplet stationary. So that must mean at this point, the rate at which he's drawing fluid out is equal to the rate at which fluid is being supplied here at the proximal tubule. So it's a way of measuring the flow rate at that point in the proximal tubule by holding this little oil droplet stationary. And this measurement here we call VTF, which is essentially the flow rate of the tubule fluid at this point. And then what you can do is you can measure, so that's another thing we measure, the flow rate in the tubule at that point. And you can also then analyze the concentration of inulin in the material you're taking out here, which is the tubule inulin concentration. So these are the things that, that he can measure. And then the question is what does this give you? Well, actually, it's a really important result. Because if all the fluid that's coming out across these capillaries is called SNGFR, so SNGFR is all the fluid coming out through filtration across these capillaries, we can do a mass balance. SNGFR times the concentration of inulin in the plasma, because that's what's being filtered, must equal what? Thinking about what he's drawn out here to make a mass balance. We have inulin coming across, and we're pulling inulin out here. So we're going to make a mass balance on inulin. So this is the amount of inulin that's being filtered across the capillary walls. What's that equal to? Can someone tell me? From the measurements that have been made? I've used this. I'm trying to compute this. Do you see anything else here that was measured? So if I take VTF, which is the flow rate of the tubule fluid, and multiply it times what? The concentration. That's the mass balance on inulin. Now, what do I know? I know this, and I know this. So what can I calculate?
It's SNGFR.
I can calculate SNGFR from these experiments. So this was a huge breakthrough. This is the first time that the filtration rate in a single glomerulus had ever been measured. Now, there's something else that he can measure, or he was measuring. In this blood, the systemic blood that he drew out to measure the plasma concentration of inulin, he can also measure something else called Ca. And Ca is the protein concentration in the blood, in your systemic circulation. And in these mice, in fact, or in rats, and it's actually in us too, it's about 6 gram%. Now this is protein's primarily albumin, but it also includes globulin, immunoglobulin. It's primarily albumin. So there's an assay that measures total protein that you can set up in your laboratory. And gram% is the same units as grams per 100 ml. Now, the other thing that he can measure is he could not stick a pipette in these capillaries and withdraw blood. The reason is what do you think happens at the tip of the pipette if I try to draw blood up through a 1.5 micron opening?
Red cell clogs it.
Yeah, the red cells just clog it. But you can take a pipette with a larger diameter, and you can puncture the efferent arteriole, which is a larger structure. And you can actually pull out blood. And in it, you can measure CE, which is the efferent plasma concentration. And he found that that was 9 gram per cent. Why is it 9 and not 4? What's happening in the capillaries?
Is it water?
The plasma is being filtered in the capillaries, leaving behind what? What's left behind?
Protein.
The protein. So it gets concentrated, just the same way inulin was getting concentrated in this structure. And you can see it went from 6 to 9 gram per cent because of the fact you had this SNGFR. You were filtering out some of the fluid in which the proteins resided. And with that, you can actually calculate another number, which is SNFF. This is what Bill Dean put on his license plate after we crashed his first VW. Because this means single nephron filtration fraction. Okay, so FF stands for filtration fraction. That is the fraction of fluid which is coming in, and since you're filtering plasma, we'll call this QA. QA being the flow rate of plasma coming into the glomerulus. That's equal to 1 minus CA over CE. Now, why does that make sense? If I didn't filter anything, what would SNFF be? What would be 0? Right, we held up a 0. If I filtered out all the fluid, CE would approach what? I mean, you know, these tapes. It would, CE would it get really big if I start taking out all the fluid in the glomerulus, filtering it all out here? CE would get larger and larger and larger. SN and F would approach what? 1. So those are the limits. SNFF varies between 0 and 1. But since I know CA and CE, I can plug in 6 over 9 into here. And this turns out to be equal to 0.33. So in these animals that he was working on, about a third of the plasma coming in was being filtered with a filtration fraction of 0.33. Now, what can I do with that number? Well, one of the things that I want to be able to measure, because it's probably important to the dynamics of this process, is what is the plasma flow rate entering a typical glomerulus? Well, it turns out by having SNGFR and SNFF, I can calculate it formally because, let me do it here. SNGFR is just equal to the plasma flow rate times SNFF. Now, does that make sense? If it all got filtered, we found out SNFF approaches what? What was the limit? 1. If SNFF becomes 1, SNGFR becomes equal to the plasma flow rate. That says that all the plasma that came in got filtered. On the other hand, if SNFF is 0, because CA equals CE, so nothing got filtered, SNFF goes to 0, and SNGFR there in QA is what? Well, you know, it's like you're dividing by 0. So if SNFF is 0, then SNGFR must have been 0. Nothing filtered out. But fortunately, I can actually solve this, right? I can solve this for QA, which is equal to SNGFR divided by SNFF. So this is an experimentally determined number now. So by having done this experiment with these micropipettes, measuring the inulin concentration in two places, the volumetric flow rate coming out of the tubule where I've punctured it, these two protein concentrations, I'm able to calculate the plasma flow rate into a single glomerulus. Now, the other thing I said is that he could measure PBS and PGC. The pressure in the glomerular capillary, that turned out to be 45 millimeters of mercury. And the pressure in the Bowman space turned out to be 10 millimeters of mercury. He gave us those data, and he basically said, go away and figure out how this glomerulus is working. So that was the engineering challenge. So what we have here is a situation where we have a capillary, a tube, and we have fluid coming in. And what we'll do is we'll model this capillary bed as just a single tube over which SNGFR comes out, and then fluid comes out the other end. And so my question to you would be, what is causing fluid to leave the glomerular capillary? What physical forces must be present to cause fluid to leave the capillary? Pressure. All right. So let's sort of draw a diagram. If this is length L of a glomerular capillary, let's just draw a diagram where we try and figure out from the afferent arteriole, which is at the front end, to the efferent arteriole, which is the other end. Let's sort of schematically plot pressure units in millimeters of mercury on this axis. And you would think that you see the pressure in the glomerular capillary is higher than it is in the Bowman space. The Bowman space is out here, and the glomerular capillary is here. So let's plot the pressure so it's the pressure difference, isn't it? It's the pressure difference between the glomerular capillary and the Bowman space. And what is that? What did he measure it to be? What's the pressure difference across that wall? 35 millimeters of mercury. 35. Now, the other thing he found is he found that no matter where he punctured this and measured pressure, he, for the most part, with an experimental error, always got 45. And so this capillary was pretty unique in that it looked like there was very, very little pressure drop from one end of it to the other. For all intents and purposes, the delta P, as we call it, the pressure difference across the capillary wall, was about 35 millimeters of mercury. Now, is there another force involved here that controls how much fluid's going to leave as you move down this capillary? You got to create a force. One is pressure. What other kinds of pressures can you think of in fluids? In fluids that have macromolecules in them, like proteins. Osmotic pressure. Osmotic pressure, good. So if I can go into the laboratory and I can get an osmometer and I can measure osmotic pressure, pi, in millimeters of mercury as a function of protein concentration. And by the way, if you're a little rusty on osmotic pressure, so how do you get fresh water on the Love Boat? By reverse osmosis. So how does that work? Well, you take a pot, you put a semi-permeable membrane in this pot, and you put salt water on this side. You put a big piston here and you press on it. And this membrane is permeable to water but not to salt. And if I press on it hard enough, what happens? It pushes the water over here, and you get fresh water. If I take this same pot and I take the piston away, what happens? The water moves from right to left to dilute the chemical potential of this salt solution. And that's osmotic pressure. So osmotic pressure is a true pressure driven by a chemical potential created by a concentration difference. So here what we have is we have protein in the glomerular capillary. And how much protein do we have in the Bowman space in that fluid? What do we know about these glomerular capillaries? How much protein gets across? 0. These are not leaky, unless you're sick, these are not leaky to proteins. So what happens to the protein concentration as I move down the capillary? It goes from 6 to 9 gram %. Well, I can go into the laboratory and I can make up a solution of protein solution that's 6 gram %. And I find out if I come over here and put it in my osmometer that I get about 20 millimeters of mercury. And if I put another one in that's about 9, I put it in my osmometer, and I get over here and it's about 35 millimeters of mercury. And in fact, I didn't do a very good job there. In fact, I can go in and I can make a bunch of solutions and I can get a curve. And I can fit an equation to this curve, which is a quadratic, which allows me to convert plasma protein concentration to osmotic pressure by getting A1 and A2 as fitting parameters to that curve. So that's something I know. I can go into the laboratory and I can measure it. So in terms of the osmotic pressure, if I say at any point X along this capillary, I have some flow of fluid across this capillary wall. Now, and I'll call it JV at X at any point. Now, this has units of flux. So this is a flux of fluid. So it would have what units if it's a flux of fluid? Gallons per area per time, right? Or mLs per area per time. That's a flux. Now, what's that equal to? Well, I do know that it must be equal to the pressure in the glomerular capillary minus the pressure in the Bowman space. That's certainly causing a flux of fluid out. And we also know that the osmotic pressure difference must also contribute to that. But what's the sign here? Plus or minus? Think about it. I'm going down the capillary and the proteins are getting more and more concentrated. What's that going to tend to want to do? Osmotic pressure wise. If the solute increases as I go down here, it's going to pull water back in. So this has to be a minus sign. And to make this an equality, I'll put a coefficient of proportionality out there. I'll call that the hydraulic permeability. And this is a property of the what? The membrane, the capillary wall. So what is pi bs? How much is that? How much protein is there in the Bowman space? Huh? How much protein is there in the Bowman space? You know, whenever you get asked these questions, it's probably either 0 or infinity. 0. All right, 0. Right? There's no protein there. So what does this say? This says that the flux at any point is equal to the difference between the hydraulic pressure driving force minus the osmotic pressure, which is trying to pull the fluid back. And they tend to compete against one another. Now, help me here. What is delta pi? What's the value of delta pi at the beginning of the afferent arteriole when it's coming in here? Delta pi is pi GC minus pi BS, but measured here at the afferent arteriole. What's the protein concentration? 6. What's the osmotic pressure it corresponds to it? 20. So that's 20. What is the delta pi at the end? 35. So, now I have to connect. You see, this was easy to connect. Now I have to connect this, right? These 2 points. The fluid comes in. So do you think this is a good way of doing it? No. Okay, that's very good. Okay, how about this one? Why? Should be constantly increasing. Okay. How about that? Does that work? Does that work for delta pi? Is that consistent with the data? Starts here and it finishes there. Looks pretty consistent to me. What would you prefer? You were drawing a different line?
Kind of an upward exponential.
How about like this? Does that fit the data? Yeah? Well, sure it does. It just says that I ran out of delta pi at this point. Well, okay, so does that fit the data? See, these all fit the data. So now we've got a problem. We can't figure out what this curve is with the data set that we have right now. And so you start scratching your head and say, well, how do I solve that problem? Well, what you do is you say, let's apply a simple mathematical model and see if we can figure out which of those curves is really the one that the animal's operating under. The animal doesn't choose which one it's operating, and the animal just is there. It's on one of those, all right? Because the animal didn't take E20. So this is just the flux at some point along the capillary. So if I wanted to get SNGFR, what do I have to do to all these point fluxes? What would you do? What urge? Yeah, integrate what? Integrate JV of x, right? So let's integrate. So SNGFR must equal the integral over the surface area of our capillaries times JV of x ds, where ds is a little incremental area element around our tube. And this formally is equal to big Kf times what I'll call delta p bar minus delta pi bar. Because what I'm going to do is integrate this equation down the length of the capillary, where Kf now is equal to little k times S, and this is called the ultrafiltration coefficient. And delta p bar is just the integral of delta p along the capillary. But fundamentally, delta p bar is really how much? Look at the picture. I integrate 35 to 35. What do you think it is? It's 35, right? Now, delta, how do I integrate this? What I'm going to have to integrate is, I can't integrate it if I don't know the curve, right? If I don't know that function for delta pi, I can't really do this integration. So this turns out to be still a question mark. Kf, the ultrafiltration coefficient, is K times S. And really what I would like to do, and the reason I'm doing this, is we know we can measure SNGFR. I already showed you how we do that. We know what delta P bar is. It's 35. And we don't know what delta pi bar is because we don't know what curve we're on. And KF is something we'd also like to get out of the data because this is a physiologic parameter. When people get sick and their glomeruli go haywire, one of the things that changes is either the surface area or the hydraulic permeability or both. That gives you an indication of disease. So if I asked you now what the determinants of SNGFR are, what would you say they are? Here's SNGFR. What controls it? Just pick them out. What's the first one there that controls it? KF. What's the next one? Delta P bar. What's the next one? Delta pi bar. And you might say, well, I'm done. I answered the question, but the problem is, I don't know what delta pi bar is. So, some engineer might come along and say the following. Some engineer might come along and say, well, what you should do is just assume it's a straight line. Just assume delta pi is a straight line, and if we do that, then we just say that delta pi is equal to pi delta pi A plus delta pi E over 2. Make it a straight line. Take an average. But if we did that and we wanted to compute KF once we know SNGFR measured, would we be calculating an upper bound or a lower bound for KF? I'm putting a number in here. Imagine if I picked one of these other curves. Is delta pi bar for this curve bigger or smaller than this one? The average is bigger. So if anything, I'm putting in a minimal value with this assumption of delta pi bar. If I put a minimal value of this in, I'm going to calculate what? For this? A minimum. So the fact is I would compute a value which is too low, a lower bound. If I put in a larger delta pi, which would give me a smaller driving force, this would have to get bigger. So I could calculate a minimum, but that's not what I want. I want to do the real thing. And so what do? You're a good chemical engineer. You go in and you make a mass balance along this capillary from left to right. And to do that, you just simply say, well, the rate of change of flow rate along this capillary is equal to minus JV of x, how much fluid leaves it, times the area. So this is now a flow rate divided by the length of the capillary. That's surface area per length. You'll see that these units agree. And the QDX is the rate at which the plasma flow rate changes. And why does it change? It changes because you're losing fluid. In fact, it drops. This has an initial condition which says that the flow rate coming into the capillary at x equals 0 is QA. So that's mass or volume conservation. What other thing has to be conserved along that capillary? What's in it we're not losing, but it has to be conserved? Protein. So you can make a protein mass balance. And that's easy. It says that dCq dx, C times Q is what? Look at the units. It's going to be mass per unit time of protein and how it changes along the length. How does it change along the length? It doesn't. So this is equal to what? This is equal to 0. So that's the other proof. And what's the condition that drives this? Well, Qx at Cx is equal to Qa Ca at x equal to 0. Now, I'll leave it to you. You can take these two equations, you can combine them, and when you do that, you'll get a differential equation. And you should be able to do this, so leave some room in your notes. Which looks like this. And so what did I do? For JV of x, I plugged in the equation right here, k times delta p minus delta pi. Combine these two equations. And what I end up with is a differential equation for C. That is the protein concentration along the capillary. If I can calculate the protein concentration along the capillary using the quadratic relation up here, what can I calculate along the capillary then? If I can solve this for C along the capillary, I must know what along the capillary then? Pi, and that's the curve we're looking for. Okay, so that's why we're doing this. We're going to calculate what that curve is. Now, if you're a good chemical engineer, you take this equation and you do what to it before you integrate it? Non-dimensionalize it. So let's non-dimensionalize the distance with respect to the length, and let's non-dimensionalize the concentration with respect to the afferent protein concentration coming in. And when you do that, and I'm going to leave out some steps because I want you to fill them in, you're going to get an equation that looks like this. So I now have a dimensionless equation. It has a parameter in it, f, which is kf delta p bar over qa. That's what must be the dimensions of f? What are the dimensions of these things? They're all dimensionless, so f must be what? This has no dimensions. So by non-dimensionalizing the equation, I've come up with one parameter, it's parameter f. And these other parameters, and these come from the quadratic A1 and A2. Delta P bar is the constant pressure difference. So all I have to do is integrate this equation. But to do that, to integrate it, I have to pick values of A1 and A2. But I know Ca and delta P bar, don't I? And I know Ca and delta P bar. So A2 and A1 are given. The only thing I don't know is F. And so I can integrate this equation for different values of this parameter F. And that's what's shown in your notes. And that graph at the top is a graph of having integrated that equation for various values of f. So you have f equals 6, 4, 3, 2, 1.5. You just put that in the computer and integrate it. And on the left-hand side is the transmembrane pressure difference across the capillary. That's the same graph I had up there versus dimensionless distance along the capillary. Now the question is what F value is the animal operating on? Well, it could be F equals 6 or 4 or 3. They all fit the curve because those curves come up and hit the delta P curve, just like we drew earlier. So how do I get a unique value of F in an animal? What would I have to do to the end of that curve? I don't want it to come up and hit delta P. I want it to come down below delta P and hit a unique value on the right-hand side. If it has a unique value on the right-hand side, then there's a unique F curve, and that's how the animal's operating. So look at what F is. It's Kf delta P over Qa. What can I do to the animal to change F and get down onto a F curve that's low? What would you do? What would you do to Kf if you could do anything? Would you make it bigger or smaller? You'd make it smaller. If I could control delta P, what would I do? Make it smaller. If I could control Qa, what would I do? Make it bigger. And that's exactly what we did. If you look at the table, we loaded the animal with plasma that increased the flow rate. It caused the value of CE to drop so that it pulled away from 35 millimeters of mercury down to something less, and we could then get an actual value of Kf, shown there as 0.078. Then what we did is that should work for any Qa that's on and that's off of having the delta pi curve hit the delta P curve. So we not only plasma loaded it, we constricted the aorta. All right? That cut the Qa down. We got 0.077. Then we took the plasma loaded animal and we occluded its carotid artery, forced more blood down into the kidney. The flow rate went up to 309, and look, Kf was 0.079. See, it doesn't matter where the animal's operating on those F curves as long as you get it away from that point where delta pi and delta P come together. Once you push that curve down, you get unique endpoints. And this was the first time that Kf, the ultrafiltration coefficient, was ever calculated. And then, remember that's K times S, the surface area? Well, then what you can do is you go in and you can take one of those molds of the animal, measure the surface area, and look at the glomerular capillary, the hydraulic conductivity of the capillaries in your kidney. It's 2.5 in those units. And look at the Ks for all the other capillary systems that have been measured in the body. They're orders of magnitude less. Nature has produced a capillary bed that has a very, very high hydraulic conductivity by at least 2 orders of magnitude in the kidney. Nobody knew that before. Nobody had understood that. And why would you do that? Well, it makes sense, doesn't it? Those capillaries are in the business of doing what? Filtering fluid. And so now you have the ability to study animals in disease states, make these same kinds of measurements, and begin to understand what contributes to the disease progression. And down in the lower right-hand corner, you might want to look at those graphs, because what they do is they now have shown us that the determinants of GFR are Kf, delta P bar, and now you see what the determinants of delta pi bar are. They're Ca and Qa, because that's what's in the equation. Ca and Qa were in this equation that we calculated from F. Now, F also had in it delta P bar, but we already picked that up. It had Kf in it. But in addition, it had Qa and Ca. So it turns out that the determinants of glomerular ultrafiltration are Kf, delta P bar, Ca, and Qa. And that's the answer. And from that, you now can say, if you come in and you're sick, what are the things I can do to change your glomerular filtration rate, in the clinic? I can change the pressure, I can change your protein concentration, and I can change your plasma flow rate. And I know exactly how your kidney will respond. So that's sort of the magic of chemical engineering applied to an important biomedical problem.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.